I am based in the Department of Zoology in Oxford, and this is their website if you want to have a look. We've got lots of information on what you'd study if you were to come as an undergraduate and the things that we do. My research group within the department is called the Evolutionary Ecology of Infectious Disease Research Group, which kind of might sound a bit weird. Well, disease, isn't that supposed to be medicine? Well, the reason we study disease within the zoology department is because you can also approach infectious disease from an ecological and evolutionary angle, and that often yields insights which you can then exploit actually for medical purposes, as I hope to illustrate to you during the course of this talk. Sorry, you know what, it's asking me for my, okay, no, maybe it will let me do that. Okay, so, start at the beginning. That's me, about 53 years ago, with my father. I was born in Calcutta, and this is a picture of Calcutta taken from probably a couple of decades before that. So I was born in Calcutta, and I spent some of my early childhood in Africa, but returned to Calcutta when I was 11 and I went to, at your age, that's where I was. I spent what I call my formative years, and perhaps you'll think later of these as your formative years in Calcutta. So Calcutta, when I was growing up there, was a very vibrant city. I'd like to say it still is, but really I haven't lived there for over 30 years, so, and things have changed a lot. But at the time, it was really dominated by a sense of the value of culture was recognized at every level, at every level of society, and within everyone was really inspired to engage with the cultural life of the city. So we had, no, that's not something I expected.
It's back.
It's back. Okay, so we had a lot of excitement about art and cinema, and everyone read a lot and wrote a lot. And so within the experimental theatre, I mean, and my father was very active, particularly in all of these fields, even though he was a professor of history at the university, but he had, the arts was sort of his passion. And so I became very inducted into that and started writing short stories and poetry and that sort of thing. And that's what I kind of saw myself doing when I grew up. It was actually what I really wanted to be was a filmmaker, in fact. And that's really how I saw myself developing. And then suddenly, about the age of 14, before which I really had not much interest in maths, a little bit in science, but more at the level of, natural history, I suddenly completely fell in love with mathematics and the science that mathematics informs, such as physics, and eventually, as you'll see, it circles back to my interest in natural history. So I became suddenly, and my father found this really odd. He said, "What are you doing? Why are you so interested in science?" And I had to explain to him that I found science actually very poetic. So, for example, this woman here is wearing a T-shirt which sports Maxwell's equations, which are a set of equations that describe electromagnetism, and they, honestly, to me, are as beautiful as any piece of poetry. And clearly this woman really thinks so, because she's had them tattooed onto her back. I would not recommend that. So alongside, so I've already got this sort of divide at this point between a sort of literature that I'm really keen and cinema and the sciences, but also within the sciences there were the mathematical, physical sciences and the biological sciences, natural history being an old love, and also molecular biology starting to become a passion of mine. So with all this in my head, I went to Princeton University, not sure exactly what I would study, but aware that the liberal arts education that it offered would allow me to pursue several different topics at the same time. And there I was fortunate to be able to do this. I went initially to study physics, to major in physics, but then I found that you could use mathematics to understand biological systems. And there I was very fortunate to have as a mentor Robert May. And essentially that's developed into what I do now within the zoology department, which is using mathematics to try and understand biological systems. And at the same time I was able to take courses and seminars in creative writing and painting and things like that. And creative writing particularly I'm indebted very much to Joyce Carol Oates, who is an American author, very prolific authoress who just turned 80. And so I was able to develop that side of my interest as well, and alongside doing, which has enabled me to pursue this other line of inquiry that I have, which is to use fiction. So I've written 5 novels so far, and what I'm doing there is trying to use fiction, which to me is another language like mathematics, fiction, poetry, to try and make sense of the human condition. So I'm just trying to show you the sort of parallels, as it were, between the two things that I do, and how, for me, that they come from the same kind of general kind of desire to understand, or at least, kind of make some accommodation for the universe that we live in, what's external and also what's within us. So I'm going to spend the rest of the talk just describing a little bit what I do on the science side, and show how this business of using mathematics to understand biological systems can sometimes have very practical outcomes, although I would like to stress as well that to me that is not the entire justification for any kind of scientific inquiry. I think scientific inquiry stands alone as a very useful and pleasurable thing to be engaged in. So I work on infectious diseases. So infectious diseases are caused by pathogens, and these range from things like viruses, shown there, that's the influenza virus there, up here, bacteria like Streptococcus pneumoniae, the malaria parasite here shown within the red blood cell where it spends a considerable part of its lifetime, and even such things that one can see very clearly with the naked eye, such as worms. That's a filarial worm, which is transmitted by mosquitoes, so it's not actually a very big worm, and causes filariasis. So what I'm going to do now is use a cartoon to kind of get across give you a flavour for what we do. So I'm going to ask you to try and think of the pathogen as this creature dressed in a particular kind of outfit. So that's now a stand-in for any of these bugs. And what happens when within a sort of an ecological system of infectious disease transmission is that people become infected by these bugs, and of course while you're infected you might suffer symptoms of disease, and then people recover. An individual will recover and become often immune. So those little soldiers are supposed to represent your immune system. So this is the sort of, at an individual level, what happens when, what infectious diseases do. So it's an ecological interaction between what we call the host, that is us, and the bug, two different species. And the purpose of vaccination is really to try and get us from being susceptible to the bug to becoming immune, having those immune defenses, but circumventing that state where you're infected and maybe diseased and even die. How is this achieved? Well, one possibility, and this is the current influenza vaccine is an example of this, is where you take the bug and you kill it. You kill the bug and then you stick it into a syringe and you deliver that to the susceptible host, and that will allow us to recognize this, allow the host to develop immunity against the bug. You don't have to kill the bug, you can just attenuate it, weaken it somehow. And this is the basis of many vaccines, such as the measles vaccine. And again, you stick the kind of weakened bugs into the syringe, stick it into someone's arm, and they develop immunity without becoming ill from the bug. You can even, this is a sort of new generation of vaccines, you can even just take those bits of clothing off the bug, stick them in the syringe, shove them into someone's arm, and they will develop immunity, in this case quite obviously without having the full bug inside them and therefore not becoming diseased. What this tells you, this particular mode of vaccination tells you then, is what those clothes are a stand-in for. What those clothes represent are literally what our immune system actually sees. When you get infected by a bug, you don't see the whole bug. What you see are parts of it. Those parts are often known as epitopes. So that's what this wardrobe of the bug is meant in this metaphor to represent. The bits that the immune system actually sees. And because of this, you can just stick those clothes in a vaccine, or you can do things to the bug itself. As long as the clothes are intact, we will mount an immune response and become protected. That is all very well when the wardrobe of the pathogen is limited, such as it is with measles. So measles has, as it were, a sort of single outfit, and all we need to do with measles is use one of those techniques to make sure that the immune system sees those elements of its that outfit, and then you're protected for life. And that's why also when you get measles, natural infection measles, you develop lifelong immunity. This is not true of many other bugs, and these are the bugs, not surprisingly, for which we haven't been able to develop good vaccines, or the sort of vaccine where you just get it as a child and are protected have lifelong protection. And one of these, one such pathogen is the influenza virus. So the influenza virus has a very diverse wardrobe, by which I mean it is able to mutate, it's able to change the bits that it presents to the immune system, meaning that when you make a vaccine, that vaccine doesn't always protect you the next time the flu virus changes. So the conventional idea of what the wardrobe of the influenza virus looks like is a bit like this. The idea is that it has a large number of hats, and essentially most of our immune response is targeted against, recognizes those hats. And so what happens when you have a flu epidemic is you make a vaccine against the current strain. So you put those hats, or if you take the virus and kill it, it sees, and people who are vaccinated will develop immunity against, let's say, the purple hat. But then the virus goes away and changes and puts on a green hat, and then of course you become susceptible again. And then you have to make another vaccine which is directed against the green hats. So that's currently where we're at with influenza. We have to update the vaccine every 3 to 4 years. People are working on a different strategy to make a vaccine for influenza that will protect against all of these strains, and they're doing that by targeting something we might call its SOX, in that influenza appears to have some parts that it presents to the immune system which don't change very much, but the problem with those parts is, just like SOX, they are actually quite hidden. So to make a vaccine that actually recognizes the SOX is a very tall order. So at the moment we're stuck with trying to anticipate what hat it's going to wear next and change the vaccine. Now, about 10 years ago, we developed a mathematical model for the evolution of influenza. And what that model suggested, to cut a long story short, is that actually people were mistaken about the wardrobe of influenza. That in addition to those myriad hats and the sort of one pair of type of socks, there were actually elements of the wardrobe that were limited in their diversity, meaning that there are parts of the flu virus that get presented and elicit a response from the immune system, which are actually not able to, they can change, but they are limited in what they can mutate to. So it only has 4 T-shirts, for example. And very fortunately for me, so this was theory we published about more than 10 years ago, 11 years ago, and nobody believed us. Absolutely nobody believed us. They literally laughed at us. So I hired someone, a postdoc, to try and test this theory, to do some laboratory experiments that would show that the virus really only had these 4 T-shirts. And what he was able to do, Craig Thompson, was actually not just do the experiments that showed that the model was right, but take it further and find the region of the flu virus which corresponds to these 4 T-shirts. And that's a picture of this epitope that he found, and that's a picture of where it's located on a molecule called hemagglutinin, which is represented by those spikes that you see coming out of that picture of the flu virus up there in the right-hand corner. So this epitope has now been identified. We've christened it Oreo after a rabbit of mine, which unfortunately no longer alive. So, but Oreo now is so Craig went further to take this epitope and make vaccines, potential vaccines out of it, and test it in mice. And so we're now at a stage where we actually have a novel approach towards making we have a patent for a new universal influenza vaccine, which will consist of the 4 different T-shirts, all stuffed into one syringe. And the beauty of that is that once you're vaccinated with those, flu doesn't have any other option, doesn't have any more T-shirts. It might have lots of other hats and whatever else, but it doesn't have any more T-shirts. So basically that's going to leave, we hope, the flu virus all dressed up but nowhere to go. So we're very excited about this. And as I said, the patent application has been filed, and we now have some people, some investors actually interested in giving us money to develop this. And hopefully we will end up, as a result, with a universal flu vaccine and have outwitted this virus, which has a diverse wardrobe, but so that ends up all dressed up, but with nowhere to go. Thank you very much.